So for this week's lab, we were to diagram and discuss the light reactions and Calvin cycle. So here's my diagrams. Here first we have the light reactions, which occur within the thylakoid space membrane, and in the stroma. So electrons are supplied to photosystem 2 by the splitting of water that gives off 4 electrons from 2 molecules of water, producing oxygen as a byproduct, as seen in this first photosystem 2 complex. Here, light is harnessed in the chlorophyll reaction center, just these little boxes up here. It causes electrons to gain energy. The absorption of light causes chlorophyll to be excited, and an electron is passed through the mobile electron carrier plastoquinone, which is the little Q. The plastoquinone powers the pumping of protons by the cytochrome B6F complex. So electrons travel from the plastoquinone into the cytochrome 6B complex, which gives it energy to pump hydrogen ions through, causing a proton gradient. So as it goes up, proton gradient is formed. The electrons are then donated to the electron carrier of plastocyanin, which is the little PC up in the top, which transport the electron to the reaction center of photosystem 1, which an addition of light, so it receives more light and excites the electrons. These electrons are then used to generate NADPH, as these electrons pass through photosystem 1, they are introduced to this little system over here called ferredoxin. Ferredoxin then from ferredoxin, the electrons travel to the ferredoxin NADP reductase, in which NADP plus is reduced to form NADPH, which forms all the energy within the cell. As these protons are transported through the proton gradient, it gives the energy needed to ATP synthase to form ATP. So NADP is given the last phosphate through the power produced from the proton gradient. And how it links to the Calvin cycle is that the ATP and NADH produced by photosynthesis is used by carbon fixation to convert CO2 to sugar, which is what occurs in the Calvin cycle, or carbon fixation cycle. So here you'll see, we'll start from the top. Here you'll see that CO2 combines with ribulose, which is through the addition of RUBP carboxylase and oxidase to form Rubisco, which is Rubisco. That is used to form 3 phosphoglycerates, which then uses 6 ATP to convert it to 6 molecules of 1,3-biphosphoglycerate, which then uses 6 NADHs to form 6 glyceraldehyde 3-phosphate molecules. From there, 1 molecule is given off to form glucose, fats, and amino acids, and the other 5 molecules enter back into the cycle, and the cycle is completed 6 times. So it enters back into the cycle. As you see, the 5 glyceraldehyde 3-phosphates, it gives off 2 more phosphates. It uses 3 ATP energies to form three ATP energies to form three ribulose 1,5 biphosphate which is a 5 carbon, and then it just continues the cycle two more times for a total of 3 times. For every 3 molecules of CO2 used, 1 molecule of glyceraldehyde 3-phosphate is produced, and 9 molecules of ATP and 6 molecules of NADPH are consumed to form this cycle.